Throughout this video, we're going to explore the phases of a cell's life cycle, from interphase through to mitosis. By the end of this video, you should be able to identify the stages and understand the basic functions that occur within each phase. Within your body, millions of cells are dividing every second. Biologists call this cellular division. Cellular division occurs to replace damaged or lost cells, cellular growth, and reproduction that doesn't require fertilization of an egg via sperm. Most of a cell's life is spent in interphase. During interphase, a cell will undergo growth and DNA replication. During the G1 phase, cellular growth occurs. Then, during the S phase, the synthesis and replication of DNA occurs. And during the G2 phase, further cellular growth occurs. Most of the cells in the human body enter mitosis. Mitosis is where cell division occurs and only takes up 10% of a cell's life cycle. However, some cells, for example muscular and nerve cells, don't divide and therefore enter into a G0 phase, never entering mitosis. For cells that do enter mitosis, the whole process from interphase to mitosis is a cyclical process, which is just a scientific word to indicate that this process continues cycling through the entire life of a cell. Almost all genes in a eukaryotic cell are located on chromosomes contained within the cell nucleus. Since mitosis is the biological process of identical cellular replication, and DNA replication is the star in this biological performance, let's focus on the structure of a chromosome first. Each chromosome contains one long DNA molecule, the genetic instructional manual for cellular function. During interphase, chromosomes are uncoiled masses of thin-fibered DNA, contained within the nucleus of a cell. DNA remains as this thin fibered mass for most of the cell's life cycle. However, during mitosis, DNA is folded and coiled with the assistance of proteins such as histons, forming compact chromosomes. This allows for easier transport of the duplicated DNA. A pair of chromosomes contains sister chromatids and a centromere. Let's do a quick recap. Cells spend 90% of the cellular life cycle in interphase, where growth and DNA replication takes place. The other 10% of a cell's life cycle is spent in mitosis, cellular division, which aids in replacement, growth, and asexual reproduction. Chromosomes contain specialized proteins and identically replicated DNA. A chromatid is the identical copy of DNA, and the centromere is the attachment point of the microtubules during mitosis. Now pause this video and see how you go with the following questions. During prophase, replicated DNA begins to coil into chromatin fibers within the nucleus. Subsequently, within the cytoplasm, mitotic spindles form. These mitotic spindles contain centrosomes, which are currently thought to be behind the coordination and relocation of the chromosomes through the attachment of microtubules that grow out of them. During the final stages of prophase, the nuclear envelope breaks open, allowing the microtubules of the mitotic spindles to attach to the centromere of the sister chromatids, moving the sister chromatids to the center of the cell. The mitotic spindles are now fully formed and the chromosomes are lined up in the middle of the cell. Each sister chromatid is attached to a mitotic spindle at the centromere. Each mitotic spindle pulls the chromatid towards opposite sides of the cell. This tug of war keeps the sister chromatids in the center of the cell. Additionally, microtubules free from attachment also line up between the sister chromatids. A cell enters anaphase with the sudden separation of the sister chromatids. At this point, each is now considered a daughter chromosome. The daughter chromosomes move to opposite ends of the cell as the microtubules shorten. At the same time, the microtubules that are not attached to chromosomes lengthen, elongating the cell. A cell enters telophase when the sister chromosomes reach opposite ends of the cell. The nuclear envelopes form around both the sister chromosomes, and then the chromosomes uncoil. The mitotic spindles degrade, and the division of one nucleus into two genetically identical daughter nuclei is complete. Cytokinesis, while not a part of mitosis, usually occurs with telophase. A cleavage furrow pinches the cells in two, producing two daughter cells. And then G1 phase repeats for both cells. A great way to remember the sequence of mitosis is with the acronym PMAT. Prophase, metaphase, anaphase, telophase. You are now ready to answer the following questions. Good luck.